Welcome to G Fun Facts Online. Today we are diving deep into a subject that sounds less like biology and more like medieval alchemy. Imagine taking a small patch of skin from your arm, treating it with a specific protein cocktail, traveling back in time to reset the biological clock of those cells, and then transforming them into the delicate, light-sensing tissue of the human eye. This is not science fiction. This is the current reality of regenerative medicine. For centuries, the central nervous system, specifically the eye, was considered a one-way street. Once the neurons of the retina or the clarity of the cornea were lost, they were gone forever. Humans do not regenerate like salamanders. But today we are exploring how scientists have effectively rewritten the rules of biology using induced pluripotent stem cells, or iPSCs, to turn the body's largest organ, the skin, into a reservoir for restoring sight. To understand this, we have to look at the biological time machine discovered in 2006 by Shinya Yamanaka. Before this, the dogma was that cellular development only went forward. An embryo became a heart, liver, or skin cell, and that was it. Yamanaka shattered this by introducing 4 specific genes known as October 3/4, SOX2, CMYC, and KLF4 into adult mouse skin cells. These 4 factors wipe the cell's memory clean, reverting it to an embryonic state. These are iPSCs. They are induced because we make them in a lab, and pluripotent because they have the potency to become many different things. This earned Yamanaka a Nobel Prize because it meant we no longer needed controversial embryonic stem cells. We could take a biopsy from a patient, rewind the clock, and have a limitless supply of personalized stem cells matching their own DNA. However, creating the stem cell is just the start. The real challenge is directed differentiation, or coaxing that blank slate cell to become an eye. The eye is a complex camera. You have the retina, which captures light, the retinal pigment epithelium, or RPE, which supports the retina, and the cornea, the clear front window. Developmental biologists have spent years mapping the chemical signals an embryo uses in the womb. By mimicking these signals in a petri dish, they can guide iPSCs to form an eye field and eventually an optic cup, a 3D structure that self-organizes into retinal layers. The first major application of this technology was for Age-Related Macular Degeneration, or AMD. In the wet form of this disease, blood vessels destroy the RPE, the support crew for the retina. Without the RPE, photoreceptors die. In 2014, Dr. Masayo Takahashi at the Riken Center in Japan performed a world-first clinical study. Her team took a 4mm skin biopsy from a 70-year-old woman, reprogrammed the cells into iPSCs, differentiated them into a sheet of RPE cells, and transplanted them into the patient's eye. The result was a landmark success. The cells survived, and because they were the patient's own biological material, there was no immune rejection. The degeneration halted, and the patient reported brighter vision. This proved a human could see through cells that were once part of their skin. But there was a catch: the cost and time. Making personalized cells takes nearly a year and costs hundreds of thousands of dollars. This is not scalable for millions of patients. This led to the current evolution of the science, which is allogeneic transplants. Instead of using your own cells, scientists use super donor cells from people with rare immune types that match a large percentage of the population. These can be manufactured in bulk and stored on a shelf. While patients might need mild immunosuppressants, the eye is immune privileged, meaning it tolerates transplants well. While that covers the back of the eye, let us look at the front. The cornea relies on limbal stem cells to stay clear. If these are destroyed by burns or disease, the cornea scars over, causing blindness. In November 2024, a major breakthrough was published in The Lancet by researchers at Osaka University, led by Koji Nishida. They treated 4 patients with severe corneal damage. Instead of waiting for donor corneas, they engineered corneal cell sheets from iPSCs. They removed the scar tissue from the patient's eyes and stitched these lab-grown sheets over them. The outcome was stunning. All 4 patients saw immediate improvement, and in 3, the vision cleared significantly and lasted. This was the first time iPSCs were successfully used to reconstruct the front of the eye. Now we have to talk about the moonshot, the holy grail of ophthalmology, replacing the photoreceptors themselves. In diseases like retinitis pigmentosa, the rods and cones that capture light die off. Replacing a support cell like the RPE is one thing, but replacing a neuron that needs to plug into the brain is infinitely harder. To solve this, scientists are growing retinal organoids. These are mini retinas, grown in 3D suspension, that actually develop layers of rods and cones that respond to light in a dish. In 2020, Kobe City Eye Hospital began transplanting these sheets into humans. The critical hurdle here is wiring. The new cells must grow axons and form synapses with the patient's remaining bipolar cells to send signals to the optic nerve. Animal studies suggest this handshake is possible, and human safety trials are currently underway. But is there a faster way? In 2020, a team funded by the National Eye Institute proposed direct reprogramming. The traditional stem cell method takes months. Direct reprogramming uses a cocktail of small molecule drugs to chemically bathe skin cells and morph them directly into photoreceptor-like cells, skipping the stem cell phase entirely. This takes about 10 days rather than 6 months and reduces the risk of tumor formation because the cells never enter that unstable, pluripotent state. In mice, these chemically induced cells restored pupillary reflexes. This represents a potential version 2.0 of cell therapy that is faster and cheaper. So where does this leave us? We are moving from a question of if to a question of when. The hurdles remaining are integration, ensuring high-resolution wiring for the retina, and cost, which automation and robotics will likely solve. We are also seeing the convergence of CRISPR gene editing with stem cells. For genetic blindness, we can now take skin cells, fix the mutation in the lab using CRISPR, and transplant the corrected cells back into the patient. In conclusion, we have moved from a world where blindness was a final verdict to one where it is a technical challenge. By looking at our skin, we found the potential to see the stars. That is the alchemy of the 21st century. Thanks for listening to G Fun Facts Online.